DNA replication occurs to create an identical copy of a cell's genetic material in preparation for cell division. It happens in the synthesis phase of the cell cycle. It is a semi-conservative replication where each double-stranded DNA is separated and the new strand is built onto the original. Each new DNA molecule has one strand from the original molecule and one new one. DNA replication begins at the origin of replication, a site within a chromosome where DNA unwinds, initiated by topoisomerase, which binds at the origin and moves outward, uncoiling the DNA, creating a replication fork. Eukaryotic cells have multiple ORIs on one strand of DNA, whereas prokaryotic cells have only one ORI. Topoisomerase uncoils and unwinds the DNA strand. And helicase is the second enzyme in the process of DNA replication. It binds to DNA and travels from the 5' end to the 3' end, keeping the fork moving. DNA replication proceeds outward from the ORI in opposite directions. The 5 and 3 and 3 and 5 ends are anti-parallel because this orientation allows for complementary base pairing and the double helix structure formation. The difference between 5 to 3 and 3 to 5 is in the carbon atom position. These numbers refer to the carbon atom's position on deoxyribose sugar and determine the directionality of the DNA strand. After helicase splits DNA, single-stranded binding proteins prevent two DNA strands from rebinding by attaching to the backbone of the strand to hold the replication bubble open. Without SSBs, DNA would snap back together because of the strong affinity between A and T and C and G nucleotides. AT and CG are connected through hydrogen bonds. AT has 2 bonds, whereas CG has 3, making it more stable.
After SSBs, proteins keep the DNA open, primase signals DNA polymerase III to bind. It does this by attaching RNA primers to the section of DNA in the replication bubble where DNA polymerase III will bind and begin synthesizing. DNA polymerase III then attaches to the RNA primer and covalently links free nucleotides together to form DNA strands in the 5' to 3' direction. In bacteria, it lays down 500 nucleotides per second. However, in eukaryotes, it only lays down 50 per second. Bases are added based on the complementary base pair rule, and at the replication fork, anti-parallel strands are opened, one from 5' to 3' and the other in 3' to 5'.
For the strand that runs 5' to 3' as the fork opens, DNA polymerase III can add bases continuously from the 5' end to 3' end. This is called the leading strand, which is continuous. In the other strand, DNA polymerase III synthesizes by stopping and going. Since it can only synthesize in the 5' to 3' direction, but the strand opens in the 3' to 5' direction, DNA polymerase III adds bases, stops, and waits for primase to lay more RNA bases, and then continues adding bases in the newly opened area of the bubble. This creates discontinuous fragments of DNA called Okazaki fragments. This strand is called the lagging strand.
Even though all the bases are added, some nucleotides are those of RNA from the RNA primer. DNA polymerase I removes the old RNA primer nucleotides and replaces them with DNA nucleotides. It also proofreads the bases laid down by the DNA polymerase III. After that, in the lagging strand, there are still Okazaki fragments that aren't bound together, which make a broken up DNA strand. DNA ligase joins and binds the Okazaki fragments together to make a full continuous DNA strand. It forms phosphodiester bonds.
Lastly, telomerase adds repeating bases called telomeres to the ends of each DNA strand. These bases act as protection for the coding strand only in stem or cancer cells. You can think of telomeres as the aglets on shoelaces. The aglets serve as protection for the rest of the shoelace. Without it, the shoelace, or DNA, can fall apart and unwind. When DNA strands become damaged, the cells cannot function properly. Telomeres shorten as cells age, called the half-life limit, making the cell only able to divide around 50 times before it dies. In cancer cells, telomerase is abnormally high, allowing the cells to continuously divide.